So welcome everybody. I can't remember how many years we've been doing this, but this has been a wonderful event that we hold 5 or 6 times every year. As Eli said, this is an opportunity for us to delve into someone's past, find out what makes them tick, how they ended up in the world of Parkinson's disease, and what they're doing. And obviously, during this conversation, if you have any questions, do stick them in the chat, and I will try and get to them. So our special guest today is now I call him Leonidas, but I now know that's wrong. I've got to say Leonidas. Is that right?
Pretty good, pretty
Pretty
good.
good. So I'm going to call him Leo because it's easier. But I've known Leo for many years, since actually I think the first time we really met was when we were on a European Research Council meeting together. And Leonidas arrived and I instantly made him my deputy chairman, which then gave him lots more work than me. And I was very grateful for that, and he deputized for me. But anyway, that's to one side. So Leonidas is a very distinguished neurologist, a very distinguished scientist who's also put together the program that you'll all, I'm sure, be coming to next year in Phoenix at the World Parkinson's Congress in 2026. And we'll talk a little bit about that towards the end, about what that actually involved in terms of that role, and how one decides what program actually looks like. But Leo, welcome to our Research Spotlight. It's always useful to know, what is your current position? So what are you at the moment? And then we'll go right back to where you began on.
Right. Currently, I'm chairman of the first department of neurology at the University of Athens Medical School.
Yeah,
And I'm also an affiliated investigator at the Biomedical Research Foundation of the Academy of Athens. So this is a research institute.
yeah.
But then most of the time, I'm in my academic position in the university, doing a lot of clinical, administrative, teaching
Okay.
work there.
So
But
you're based in Athens, and you have this sort of
right.
hybrid life of seeing patients, doing research, and running administrative sort of duties within the clinical school and the research institute.
Yeah, in the research institute, I have a lab, and I'm still trying to do some more basic or translational research with cell models and animal models.
Yeah.
But the truth is that my basic work now is more in the university with all these other roles I have. Yeah,
Yeah.
including, taking care of these very rare cohorts of genetic synucleinopathies, as we call them.
Okay. So go back to, if we can go back to your sort of origin. So born in Greece, grew up in Greece, did your medical
Oh,
training
that's
there?
wrong. You didn't read my CV well. Okay, I was born in Canada, actually, so I'm
Yeah.
Canadian.
Right.
Maybe the 51st state, who knows?
Okay.
But then it was still a country.
Yeah.
Yeah,
Yeah.
in 1962, I was
Yeah.
born there. And but my family moved back to Greece pretty soon after that.
Yeah.
And so I grew up in Greece. So I'm basically went to school, everything in Greece, military service even in
Okay.
Greece. Yeah,
And did you know
medical
early on
school.
you were going to become a doctor? Is that something that runs in the family, or was it
Well,
a
my
bit?
father was a psychiatrist, a very eminent psychiatrist. He was even, chairman of the World Psychiatric Association at some point,
Right.
and very distinguished also for his research. Actually,
Right.
it's very interesting because I think we talked about this. He performed research in together with Herbert Jasper at McGill
Okay.
when Penfield was doing the first surgeries for epilepsy and mapping out the brain.
So these
So
were
my
where
father was
they recorded from people's brains and stimulated and worked out how the
right.
brain is organized in
While they were doing
people.
surgery, and it was really pioneering at the
Yeah.
time.
Yeah.
And Herbert Jasper was the neurophysiologist
Right.
involved in those operations. And of course, he had a research lab, and that's where my father went. And he
Okay.
did research on electrophysiology in large animals.
Okay.
And he then went to the NIH, to a pharmacology division there, and he came back to Greece after all this research, but he ended up doing more psychiatry after this. So
Okay.
although he was very deep into neuroscience, he ended up doing more psychiatry finally.
And was
So
there
in a
ever a time
sense,
in
I
your
had a
life?
similar trajectory to him, in a
Yeah.
way. Yeah.
And was there ever a time in your life you didn't think you'd be a doctor?
Well, I wanted to do perhaps more biology. That was
Right.
my but because I don't know, it was a kind of someone I don't remember in the family told me this, that maybe because I wasn't very, outgoing and that there was a worry that I would end up, being a lab rat, not having interaction with people and just being there in the lab and not not interacting. So I ended up, I always had the interest in more basic biology, but I
Okay.
ended up doing medicine after,
Okay.
considering that. But it
And you
was
did,
between
and why did you,
biology and medicine.
and why did you choose neurology and not psychiatry, given the family tradition?
Well, as I said, my father was, as in Greece also as in other countries, it used to be a common specialty. For example, my father was trained in both neurology and psychiatry.
Right.
And actually, it's interesting because even when he came back to Greece, he was involved with both neurology and psychiatry, and he even described a series of patients. Um, uh, this is the family lore, but it's actually true that of course, Kennedy syndrome, this neuromuscular Kennedy
Yeah.
disease. He
But this
described
is
some,
this is nothing to do with Parkinson's. This is a
yeah,
rare
it's
disorder where people
neuromuscular
get
genetic
wasting of
disorder.
muscles.
And
Yeah.
he described some patients with that disorder in Greece, in a
Right.
Greek journal, even before it was published, abroad in the
Okay.
60s.
Yeah.
So he was quite, he was both a
Yeah.
neurologist and a psychiatrist. But he ended up, of course, being a chairman of psychiatry and then being very much involved in psychiatry.
Okay.
But anyway, so I was interested more in the, as I said, in the basic biology and how
Yeah.
do things work. And
Yeah.
it seemed to be that much of that in psychiatry.
Okay.
I was interested in endocrinology actually, also neuroendocrinology, how
Yeah.
the whole, organism works, but coordinated by the brain. Then I
So how
thought
hormones
maybe
work
it's
and all of
better
this. Yeah.
to
Yeah.
study the brain rather than the endocrine system. Yeah,
Okay. And so you went to medical school in Athens,
right.
and then what happens in Greece? Do you then have to do general medical training before you do neurology training, or do you go straight into neurology? And then how does we'll then
Yeah,
come on to how you got into research, given that's your great passion.
I was, I had to do the military service after
Okay.
medical school. And during that time, I sort of, in a sense, although I did 24 months of military service, it's not a
Wow.
short time, but it
That's
gave me
still two
time to
years.
think. You have a
Right.
lot of time to think when
Yeah.
you're doing your military service.
Yeah.
So and that's when I developed this idea that I wanted to go to the States and, wanted to go abroad in general. It
Yeah.
was either the States or Canada.
Yeah.
And it seemed difficult to get into the Canadian system, whereas the Americans had a much more receptive system for foreign medical graduates.
Yeah.
So I studied, for the USMLE, the exams, during my military service. So when I finished my military service, I did some further work I was doing in the lab on molecular biology, and I was, I did my interviews and stuff to really, and I was very happy to end up at Columbia
In
University,
New
which was
York.
really a dream for me. Yeah, in New York, which I loved. My sister was there at the time, actually. My
Okay. So
sister
you
was the
finished
most.
your military service and then you went straight into research in Columbia. And
No, I did. I did. I was working in the lab doing more like molecular. I just wanted to
okay.
get my feet in molecular
Okay.
biology, so I was doing some molecular biology
And
stuff.
and when you went to Columbia, how long were you there for? You were there for quite some time.
I was there, yeah. Well, essentially for 12 years, yeah.
12 years. And that involved lab-based research as well as clinical work.
Well, I was thinking, when I went to the US, I wasn't clear what I wanted to do. Should I do my clinical training first and then do more research? I even had an offer to do a kind of combined residency and PhD program
Yeah.
in the US, but I finally decided to do a residency training in neurology. So 1 year of internal medicine, which I did at St. Luke's in New York, and then 3 years of neurology training at Columbia.
Okay. Okay. And then also
As I
all of
mentioned,
your
my sister was there at the time, and
yeah.
I was
Is
going to
she a
say she's the most
neuroscientist?
eminent member of our family. She was studying visual anthropology at the time at NYU, and she became a very well-known documentary filmmaker,
Okay. Has she made
and
one of
she's
you?
great. Sorry.
Has she made a documentary of you?
No, not yet.
Well, there we
But
are.
she
It's
does
work.
these very personal documentaries.
Okay. Yeah.
It's a part of cinema verite. She really follows people for a long time and
Yeah.
gets to know them very well. They get to know her very well, and then she starts filming. And
Okay.
it's really a very personal view of situations and people.
Okay.
It's really great what she
Bit like
does.
the research spotlight.
Right, exactly. Yeah.
So
Yeah.
I mean, when you went to Columbia, did you have a particular research question or area you were interested in?
Well, I thought, neurodegenerative diseases,
Yeah.
more generally, and as I went through the residency, that's what interested me more. At the time, I was intrigued. You'll be interested to hear this. The concept of growth factors.
Okay.
What could growth factors do, and regenerate, rejuvenate the nervous system. So
Yeah.
that was kind of a topic that was of interest to
So the
me.
idea was, so this, for those who aren't familiar, this is a topic which began many years ago, but it became very big topic in the 90s and the turn of the
Right,
century. It's the idea that there are specific factors which enable nerve cells to grow, trophic factors or growth factors, and then diseases may actually be a consequence of the lack of them. And if even if not, you could treat them with it. So
right,
nerve
right.
growth factor for cholinergic cells for Alzheimer's disease, GDNF for Parkinson's, which many of you would have heard of. So you're interested in growth factors and neurodegeneration.
Right. That's what developed during my residency.
Yeah,
And, during the residency, we had elective time to do some research.
yeah,
And I really liked, also just as a person, I really liked Bob Burke.
oh, yeah.
And the research he was doing in the lab was very interesting.
So a very
Uh,
eminent scientist in Parkinson's. Well,
in,
that's a
in
minor
the field of
disorder.
Parkinson's. And
Yeah,
he was studying, um, he and Lloyd Green, which I will mention later, was studying this concept of programmed cell death in
okay.
development, which was kind of the flip side of what growth factors do. It's
Yeah,
actually what happens when you have a lack of growth factors during development. And, the cells undergo a very programmed way to degenerate
yeah,
during development. And that's a normal part of development
yeah,
because part of development is some cells survive and other cells die, which, for example,
yeah.
don't have the right connections, for example. Um, so
Which is
Lloyd
interesting because
studied,
people have often said, is neurodegeneration so a reversal of development? So, things happen
right,
in development, they grow, and then when they degenerate, they basically it's a sort of recapitulation of the or a loss of the developmental program.
right. So that was, and the biochemical pathway regulating that is apoptosis.
Yeah.
So, which is a Greek word, of course.
Okay. Like
So I
many
was
of them in
interested in
medicine.
that.
Yeah.
And Bob Burke had a nice animal model, which he had seen that he could, during development, it was a developmental model in which you would kind of cause damage to one part of the brain, the striatum, and you would have a secondary apoptotic cell death of the substantia nigra neurons, the dopaminergic neurons
Right.
that degenerate in Parkinson's. But this was in a developmental model.
Okay.
So what the
So what was
idea
the what
was,
was what was what was the lesion in the model which
the
made those
lesion was
cells think?
an excitotoxic injury, abotinic
Okay.
acid, in the striatum. So you would cause damage to the striatal neurons, and
Right.
then the substantia nigra dopaminergic neurons that project to the striatum would suffer a secondary form of
They sort
degeneration,
of
which
die
was
back and
apoptotic in
yeah.
development. So they had all the
Yeah.
morphological characteristics of apoptosis.
Yeah.
So the idea that kind of the project I did in the lab was to see what happens in adult animals when you did the same type of
Okay.
lesion. And there, it was interesting what we found. It was a cells, particular cells, not the substantia nigra pars compacta, but the reticulata neurons degenerated. But
Okay.
it was a non-apoptotic form of cell death,
So the wrong population and the wrong form of
the
cell
wrong
death.
population. But it was a very interesting. I think it's still very interesting that you can, cause damage to one part of the brain, and then
But the
secondarily
idea would be you
you can
lesion the
have
striatum, and you were hoping that this would the nigral dopamine cells would die back and you'd get an apoptotic cell
Right.
death in the nigra, but the striatum projects down to the nigra to a different bit, and
To a
it was that
different bit.
projection
Yeah.
that was actually killing off cells in the
Right.
nigra.
Right. So the forward type of
Yeah.
degeneration. Yeah. Instead of a backward type.
And so
Yeah.
where did that lead you next in your research then?
So then I was, this was a very nice study we did with Bob. As
Yeah.
I had mentioned to you, I think recently, I was so I don't know, honored when Andres Bjorklund came to my poster at the San Diego Neuroscience meeting, and he was interested, and
Yeah.
he had done some similar work. So,
Yeah.
but anyway, so it was a very nice piece of work. It kind of got me into the experimental, approaches in animals. But I felt I wanted to understand more deeply, the mechanisms underlying this at the cellular level.
Okay.
So that's, as I mentioned, Lloyd Green, who was in the Department of Pathology at Columbia, was doing work on cellular models, and where he would deprive cells of growth factors, and they would undergo this programmed cell death, apoptotic type of cell death. And so the idea was to uncover the mechanisms through
Okay.
this, through which this occurs.
And were
So
you looking
that's
at this in the dopamine cells of the midbrain, or was it a different
no,
population?
this was in actually in PC12 cells, this cell line model,
Yeah.
which is partly dopaminergic,
Yeah.
and in sympathetic neurons actually in culture.
Okay. Okay.
So the main primary, but these neurons also degenerate, as you know, in Parkinson's.
Yeah.
So they are part of the autonomic nervous system.
Yeah.
So if you deprive them of NGF, which is the growth factor they
Yeah.
rely on in culture, they undergo this program type of cell death.
Okay.
So that's what I was studying,
So how did you then pivot from that? I mean, I can see the obvious connection with Parkinson's, but when did you sort of more explicitly say, I'm now going to study Parkinson's rather than just a sort of, if you like, a mechanism of cell death in the absence of growth factors?
Yeah, I mean, eventually, of course, there were other stimuli to induce cell death, not just
Yeah.
deprivation by growth factors. And I stumbled also upon an interesting kind of concept of kind of active but non-apoptotic type of neuronal cell death. But then it was at this time that actually the story of alpha-synuclein started emerging.
Okay.
And
Which began in Greece, didn't it?
This began well, it sort of began in Italy, but it was really Greek people in Italy.
Okay.
Yeah.
So just tell us that story. I mean, so obviously, we are of a similar vintage. And when we were training, the idea that Parkinson's had a genetic basis was not really recognized. And certainly understanding what went wrong in Parkinson's was pretty limited. But this discovery of this family in the 90s, which I think was published in 97, I can't quite remember, I think it was 97.
97, yeah.
Yeah. And so, I mean, can you just sort of fill us in on what was special about that family and how did that transform our understanding of Parkinson's?
Well, the basic, I mean, idea at the time, as you mentioned, is that there's something in the environment that causes Parkinson's, and really people weren't paying much attention to familial forms of the disease.
Yeah.
But then Roger Duvoisin and Larry Gold from the University of New Jersey had, together with some Italian colleagues, Diorio is the main one, they had stumbled upon some Italian immigrant families who had many cases with Parkinson's. And they had
This
been
was in
studying
America,
them.
they
This
discovered
was in
this.
the America, yeah, in New
Okay.
Jersey, actually. And they were studying them for many years, had published some papers. But at that period, it wasn't as easy as today, to identify a gene.
No, no.
So they really had a very laborious process over many years. And eventually, they collaborated with Roger Nussbaum at the NIH to to clone, as we call it, the responsible gene. And
Yeah.
it was a disease that hit in every generation. So there were many, many members of the family. So they were finally able, with those laborious processes at the time, to hone in on the particular gene that was involved.
And so
But
these were
even
people who developed Parkinson's quite early in life, or did they
quite
develop
early in
it?
life, yeah,
Yeah.
it's in rather mean age of onset, as they had mentioned, is in the mid 40s, and that's what we've been seeing
Okay.
even now.
Okay.
And the disease is rather aggressive.
Okay.
Yeah,
So this was a
rather
sort of rather atypical form of Parkinson's in the sense that it was slightly younger and more aggressive, but
more aggressive.
but
Yeah,
the
but otherwise,
presentation was
it's pretty. I
yeah.
mean, it's pretty typical Parkinson's.
Yeah.
Sometimes there are variations, but yet generally it's quite typical Parkinson's. But just it, follows more aggressive course, especially regarding cognitive decline.
Okay. But it responds to medication and you get the
Response to
complication.
medication, yeah. The first few years, you can't really differentiate these patients
So
from
this work
patients with
then led to the
Parkinson's.
discovery of the gene which lay at the heart of it. Where does the Greek side of it come into it?
The Greek side comes from the fact that all these people that were in New Jersey, but they all came from the same village. That's why, well, town, which is called Contursi, and it's in southern Italy.
Yeah.
And but originally, this area, and actually just below Contursi, there's an ancient Greek site, which is called Paestum, and but which is called Poseidonia, which means the town of Neptune in Greek. And there was a Greek colony there, and people, in the Middle Ages, people migrated to the mountains. Essentially, that's where Contursi is, but it's right above Paestum.
Right.
And so those people are of, Greek origin because
Yeah.
that's where they came from.
Yeah.
And actually, in that original paper, it was a remarkable coincidence that the person who was doing work in Bob Nussbaum's lab was actually Greek, and his name was Michalis Polymeropoulos, and he was a graduate of the medical school in the University of Patras, which is on the western coast of Greece. Coincidentally, just that part of Greece where there was an earthquake in around 4th, 5th century BC, and many people, because of that, left and migrated to southern Italy. So remarkably, this person remembered that Michalis remembered that his professors in the university, a biologist, Aglaia Athanasiadou, and a neurologist, uh, had mentioned to him that there were these families with Parkinson's in the area, and they were studying them. So he called his biology professor, Aglaia Athanasiadou, and he asked her, what about these families? You have to send me, genetic material from these families because I think I found a gene for Parkinson's. And remarkably, these families had the same genetic defect as the Italian families.
And
So
they
that
look clinically
proves
the
they were
same.
essentially the same origin. So
Okay.
there's a founder effect at some
Okay.
point in Greece, and there's a term which is a kind of an ancient term, which is called ikistis, which means the one who sets up a colony. So
Okay.
the ikistis, the settler of the colony, was actually, transferring this mutation, presumably, from Greece to Italy and then to New Jersey.
Okay.
So it's
So it's
quite a
traced all the
remarkable
way back.
story of this
Yeah.
trajectory. And a colleague in Italy was trying to team up with archaeologists to kind of write an article about this, but
Yeah.
hasn't happened yet.
Yeah.
It's
So it's very
about
interesting. So this sort of detailed clinical observation, just noting away, recording, at the time it wasn't producing answers, but subsequently when discoveries come, you can go back and
right.
link the two together. So this was the first genetic cause of Parkinson's in
This was
our
the first genetic cause, and even highbrow geneticists thought that it wasn't clear. There were
yeah,
some articles published. You may know about this, right?
yeah,
I won't say the name, but
yeah,
we all know who it is. So anyway, there were kind of doubters about
yeah,
whether this could be, the a real genetic cause. I mean, the particular gene, alpha-synuclein, because
yeah,
it was the very interesting thing that the mutation in causing the disease in humans was actually the normal amino acid in rodents.
yeah.
So they were
We don't
questioning
get
whether this,
Parkinson's.
yeah, which don't
Yeah,
get Parkinson's. Yeah.
yeah,
So there was some doubts in the beginning
yeah,
about whether this could be the real gene responsible for the disease in these families. But what was interesting, coming back to Colombia, is that Bob Burke, as at the time they were doing these genetic studies, as I said, they were very laborious and where they were going closer and closer to the area of the gene. So they had published this paper a couple of years before the main paper, where they said, the gene responsible is in this area of the genome, but we don't know. There are many genes there. We don't know which one it is. But Bob Burke had identified the same gene, alpha-synuclein, as being upregulated in this. So it changed its expression during the same model I mentioned before, this developmental model
All right.
of apoptosis. So he was interested in this gene, and he knew it was in the area, and he actually contacted, Bob Nussbaum, and he told them, there's this gene there. I found that it's regulated in
Yeah.
dopaminergic neurons in this developmental model. Maybe you should look closer to this gene. And this
Okay.
was before it was found to be the gene.
Yeah.
Yeah.
And it's very interesting because, of course, it's so familiar now, alpha-synuclein. But if you'd
Right.
said alpha-synuclein in 1996, no one would have any
Right.
clue what you were
Right.
talking.
Right. Right.
And then I think if I remember correctly, in '98, there were people here and in the States who then showed that alpha-synuclein could also be found in Lewy bodies in people with
Yeah.
sporadic
That
disease. So it was
that came very
relevant.
soon afterwards, but it was after. So the
Yeah, yeah,
genetic discovery was the first one.
yeah,
Maria Grazia Spilantini had been working on alpha-synuclein, but
yeah,
not in relation to Parkinson's,
yeah,
as an interesting, neuronal
yeah,
gene and protein.
yeah.
So,
And
so
then
it was,
and then in Colombia, you started working on alpha-synuclein.
yeah. So the basic idea there, it's regulated during apoptosis. So does it have to do with an attempt of the cell to combat the injury it has received, or is it actually a driver of degeneration?
Okay.
So we did quite a few studies,
So you're trying to
and
bring
this,
these strands together. We've now got a genetic cause of Parkinson's. You've got Bob's observation that in development, if you knock out the system, alpha-synuclein goes up. So is it trying to compensate or is it part of the
right,
process? You've got this programmed cell death as well as this non-programmed cell death. And so I guess the question is, which I think is what you're saying, is that, is alpha-synuclein driving possibly apoptosis in Parkinson's disease in some way? That was the
right, right.
and
And so we
alpha-synuclein
did,
just, I mean, alpha-synuclein's normal, isn't it? I mean, it's a normal constituent of our brain. And so this is a big component of our brain, but the argument is in Parkinson's it somehow changes because most people with sporadic disease do not have this mutation that you were talking about.
right. It's very rare, but we know now well, we've known for many years, with post-mortem studies, but now, as I imagine many people here in the audience would know, even in living patients, you can find, if you do spinal tap and you take cerebrospinal fluid from patients, more than 95% of patients have abnormal conformations of alpha-synuclein in this biological fluid. And so it's a kind of a biomarker, right, of
Yeah.
the disease. And interestingly, it's also present in what we call prodromal Parkinson's. So
So
many years before you,
it's gone from being the preserve of a rare family coming out of Greece to
right,
to
because that was the
probably being
original
the best
discovery, yeah,
diagnostic test
that
we've got
has
for
led to
yeah,
this, to the biomarker discovery and all the interest
yeah.
and the therapeutics, of
Okay, well,
course.
we'll come on to that because I'm interested in the
Yeah.
therapeutic. So during your time in Colombia, I mean, it's hard to summarize things. I mean, what would you say was the sort of major discoveries you made about alpha-synuclein Parkinson's? Was there a sort of clear message that came out of that work?
Well, the original work was focused on these questions we talked about, and what we
Yeah.
found interestingly, and it's still something that interests me, but I haven't done much work more recently, is that actually the growth factors I mentioned before
Yeah.
induced the expression of alpha-synuclein.
Okay.
So in our cell models, for example, when you gave growth factors to PC12 cells, you upregulated alpha-synuclein. So it
Okay.
was um,
Is that a bad
the
thing?
general, and it was not regulated during apoptotic cell death in the cell models. So This meant it was more a response of the neurons, to improve the differentiation, uh, and we know it's like, alpha-synuclein regulates synapses and has a beneficial function. So it goes along this idea that, alpha-synuclein has a normal function
Yeah.
and it's regulated when it has to do some work, during differentiation and synaptogenesis.
So
So,
increasing it with growth factors isn't
so it didn't
necessarily a
link
bad thing.
alpha-synuclein per se to degeneration, these studies that we did.
Yeah.
But we created a cell model where we had the mutant form that had been described, the A53T form
Yeah.
of alpha-synuclein has been identified in these families. And there we found, and that was, I think, the main discovery, that these cells that express the mutant form of alpha-synuclein degenerated in a non-apoptotic form of cell death. But there were very interesting things about the biochemical pathways involved, and that's where what I found is that it was in relation to pathways that degrade proteins. There are two
Okay.
major systems that degrade proteins in cells: the proteasome system and the autophagy system,
Yeah.
and both seem to be affected detrimentally by mutant alpha-synuclein and not by the normal alpha-synuclein.
Okay.
So
So the argument,
that's where my
so the
interest in.
hypothesis would be that people with the mutant form, because obviously for 40 odd years they're doing all right, but the
Right,
mutant form, it's basically inhibiting the sort of collection of the garbage,
right. The
and
garbage
so over
collection.
time it sort of
Yeah.
it can't get rid of the protein it's producing, which starts to accumulate, and once it starts to accumulate, it causes problems, and that might also explain why it sort of slightly runs away with people once it begins, because it,
Right. It's kind of a vicious cycle.
yeah,
Yeah, that's the
yeah,
idea. So
yeah,
the protein itself causes damage to protein degradation
yeah,
systems. And then when they don't function well, the protein alpha-synuclein and others are not degraded sufficiently, and that causes further damage to the protein degradation systems. So this kind of vicious cycle has been, both parts of it have been the focus of my research ever since.
and so Lee, over the years, have you focused more on the mechanism by which alpha-synuclein inhibits those pathways, or or have you been looking at ways to try and upregulate those pathways to overcome
Yeah,
sort
both.
of blockage? Yeah.
Recently, more on the therapeutics angle, and we've honed in. And this was originally work actually that also started at Columbia. This
Yeah.
was work in collaboration with Ana Maria Cuervo,
Oh, yeah.
a very eminent scientist at Albert Einstein College of Medicine. So she had basically, together with Fred Dice, had invented this new degradation pathway, which was called chaperone-mediated autophagy (CMA),
Yeah.
and they had been working on it, but very few other people had been working on it. But David Seltzer, who was at Columbia,
Yeah.
got wind of this. He gets wind of all these kind of very interesting things. And he thought that maybe this pathway was related to alpha-synuclein. So
Yeah.
that's how we did a collaborative work. And indeed, we found that alpha-synuclein seemed to be degraded by this pathway. And
So can
actually,
I
that the
just pause
mutant
you at that point? So
forms.
just so we got it clear, so there are two pathways. One is a sort of protease system where you take a protein and you basically put it through a shredder and it just sort of degrades it. Whereas the sort of light, the autophagy pathways where you get whatever's abnormal, you shove it into a sort of a little package and you put it into a cell which then destroys it like a sort of acid bath, destroys it, and then it spews out the
The
bits it can.
lysosomes, yeah.
Yeah,
And
yeah,
the CMA pathway, this pathway you're talking about, is
yeah.
very selective, that the proteins have to be targeted to this pathway. And then they're brought to the lysosome and then degraded through a specific pathway.
And
And you
every
can,
single cell has this, and every single cell needs it because all the time we're producing abnormal proteins we want to get rid of, regardless of whether we have Parkinson's or not.
yeah, and the good thing about this pathway is that you can molecularly regulate it by either overexpressing or downregulating a very specific protein, which is the membrane, and does this work of threading the protein into the lysosome and causing its degradation. So that's what we started doing when I came back to Greece in various models. And this has, I mean, has taken many years, but has culminated in more recent work. We're actually doing kind of gene therapy in monkeys. And this
Okay.
is in collaboration with Erwin Bazar and Benjamin DeHay at the
Oh,
University
well,
of
anyway,
Purdue.
but Zard, he's been on one of these research spotlights, so everyone
Yeah,
here will of course know a lot about his work. But so this is in non-human primates. Uh, it's obviously based in France, but does a lot of work in China and such like. So the idea
right.
here is to try and specifically target this autophagy pathway, this one where you package it
Right.
up, put it into these lysosomes, get rid of it, but target it so it only upregulates the removal of the abnormal alpha-synuclein, or just upregulate the whole process and say it doesn't really
Yeah,
matter if other
that's
things get involved.
one of the, I mean, potential drawbacks of this approach, that
Yeah.
it's not just for abnormal alpha. It's for alpha-synuclein more generally to
Yeah.
degrade it, but also
That's
for other
normal
proteins,
alpha-synuclein. That's normal.
that are
Yeah.
degraded through this
Yeah.
pathway. So it's not selective for
Yeah.
alpha-synuclein.
Yeah.
On the other hand, we know that there are a lot of co-pathologies in
Yeah.
patients with Parkinson's disease, not just alpha-synuclein, but
Yeah.
other abnormal proteins
Yeah.
accumulate. And it's interesting that this pathway in recent work has seems to be, a potential therapeutic target in, for example, for Alzheimer's.
Okay.
Both tau and beta amyloid can be, processed through this pathway. So the idea is that perhaps by improving this pathway, you can improve more generally the proteostasis in, especially in aging brains.
Yeah.
And we know that this pathway of autophagy, CMA, but also other degradation systems, with aging, they are not functioning as well.
Yeah.
So, in a sense, it goes back to the rejuvenation
Yeah.
uh, idea, right? Trying to, improve a system that does
Yeah.
deteriorate with aging anyway.
So the
So,
advantage of this would be you could target not just any alpha-synuclein pathology in Parkinson's, but any pathology. And also from an investment point of view, people would say, well, if this works in Parkinson's, it might work in Alzheimer's, which is, 5 times more common.
right.
It could work in any
We haven't done any
neurodegenerative.
work ourselves on
No,
Alzheimer's.
but the principle is that if it works in one, it could work in all of these
Right,
conditions. So
right,
your approach is really about increasing degradation of alpha-synuclein, whereas
right.
actually most of the clinical activity in terms of trials has been in stopping the production of it.
Well, the antibodies, because antibodies are the most,
Yeah.
advanced therapeutics that have gone to the clinic. They're also, in a sense, binding alpha-synuclein and clearing it from,
Okay.
whatever bad thing it's doing. So it's not actually the production. Production would be more an antisense
Okay.
approach,
Which some people are
something like
taking.
that, which are also starting to be in the clinic, but are not as advanced at
Okay.
this
So
point.
so those immunization therapies, whether they're passive, just adding an antibody or inducing one, they haven't been very exciting, the results.
Yeah, in
Are you wrong?
2022,
Is
there
your
were
theory
two
wrong?
papers published that seemed to have negative results,
Yeah.
although in one of the two cases, therapy is still being investigated and had some interesting further results in subgroups of patients who are advancing more rapidly. These are always early stage Parkinson's disease patients.
Yeah.
So in those, there seems to be a quite considerable effect. But of course, it's kind of analysis of a subgroup of a
Yeah.
large trial. So it has to be taken with a grain of salt. But there are, within that, if we can call it failure, there are some encouraging signs that
Yeah.
perhaps this approach will eventually work.
Okay.
And there are questions about whether you are targeting the exact right forms of alpha synuclein, whether you are reaching alpha synuclein in the brain, at the right
Yeah.
areas. But still, I think, this or other approaches targeting alpha synuclein, I think eventually will be the answer. Not the answer, but, will have a disease modifying effect.
Okay. And
Uh,
so coming back to your own theory, there's also an interesting question here about autophagy is upregulated by fasting. So if you
right.
starve cells, it goes. So could we upregulate autophagy in all of our brains by
Yeah, I
just
mean,
repeating it?
fasting is a very well known, in lower organisms, it's very well known to increase survival.
Yeah.
It's certainly, it stimulates, and it's thought to be mainly through autophagy,
Yeah.
through clearing mechanism. So yeah, but of course we know, patients with Parkinson's, they have this frailty, and they have some wasting, that doesn't, it's not
So do
a
we
good
know, for
thing.
example, if you were fasting, so if I said, right, you're not going to eat for the next 4 days, do we have any idea whether autophagy would be upregulated in your brain?
I don't know if those studies have been done in humans. As I said, in lower organisms, certainly you can achieve that,
Because
and
it
you
is a
can
big
get,
problem. I mean, I do studies here with David Rubenstein, I'm
yeah,
sure you know, who works a lot on
right,
autophagy. And one of the big challenges with so we try and do trials in it, not necessarily in Parkinson's with autophagy. The big problem is we don't have a measure for it in people's brain.
right,
We can't see it. We can't measure it.
right.
Uh, you can measure proteins, but you can't actually measure the process itself. So it makes it very difficult to know whether these interventions are working through that mechanism. But coming back to your own work, so you've got a gene therapy, you were saying, is that right? Which you're using in,
Yeah, it's as I said, you overexpress this protein, which is called LAMP2A, and
yeah,
we've shown, because we're working with experimental animals, so you can show that you're upregulating this CMA pathway in the brain,
yeah,
and you get beneficial effects against insults related to alpha-synuclein
okay,
overload. You decrease aggregates of alpha-synuclein, you improve survival of dopaminergic neurons, and as I said, we're now doing studies in primates.
okay. So the plan would be that will translate to the clinic.
The plan would be, yeah, of course, a more easier way would be a small molecule, would achieve a similar effect. We have been working on that, but others have published some work related to small molecules that could activate this pathway.
Because you could give that systemically and it would affect every
You could
cell.
give that systemically.
Yeah,
And there was a very recent work by Ana Maria Cuervo's group, just very recent, showing nicely that various organs in the body, um, there's a with aging there is a substantial effect, detrimental effect in this degradation pathway with aging.
okay.
So I don't think it would be detrimental for all organs to have some activation of this pathway. But of
Yeah,
course, the main target would be the brain in this particular
yeah.
case.
Okay. So you're very much working on, I mean, the problem with the gene therapy to the brain is presumably it's only got a localized distribution. You can only inject it in a certain number of sites and it distributes over a certain area. But I guess your translation to the clinic, I know it's a little way off. I would imagine you're probably going to target the nigrostriatal dopaminergic pathway as your opening therapy. Was that where you would target your gene therapy?
Yeah, as I said, this is more of a proof of principle, but that's what we're
Okay.
doing right now. Yeah, just targeting an nigrostriatal pathway in a different kind of a strategy, which is more along the lines you mentioned earlier. We've also used some systemic, but this is in rodents.
Yeah.
We've used some systemically administered viruses, which
Yeah.
you can administer intravenously,
Yeah.
and they have good uh, brain penetration,
Yeah.
and you can achieve with systemic intravenous injection, widespread expression of the virus in the brain. So, but we're doing that actually with a virus that downregulates alpha-synuclein. So this is the production approach you mentioned before.
Okay. So this is
So
not targeting the autophagy pathway. This
this is
is
not
targeting.
targeting autophagy pathway, but potentially the same
Yeah.
kind of strategy with a systemic injection,
Yeah.
like, as you know, is available already clinically for some conditions.
Yeah.
Systemic injection of the virus that has good brain penetration.
Yeah.
In our case, downregulating alpha-synuclein or overexpressing LAMP2A to induce autophagy could be, therapeutic strategy for the future. And
And
this
they're not. And they're.
goes all over the brain. It's really
Yeah.
very nice to
Yeah.
see the expression.
And I've
And
always
we,
said
we
that they're mutually. I mean, you can imagine there's a sort of symbiosis between this. So in Huntington's disease, which is a completely different sort of a genetic, you produce the wrong protein because of the genes. But if you switch off the abnormal production of the protein and you increase its clearance, so you tweak down a little bit on the production, tweak up the clearance, the two together
Yeah.
could be a very powerful way of sort of holding the condition at bay and actually reversing it.
Yeah, and we're actually trying some models, which is the reverse of what you're saying, which
Yeah.
is perhaps what happens in, not in genetic, but in idiopathic Parkinson's, in normal Parkinson's. In normal Parkinson's, for example, there's been post-mortem studies showing that this pathway, the CMA pathway, is dysfunctional in the brains, and
Yeah.
the levels of the main proteins in this pathway correlate inversely with the accumulation of alpha-synuclein. And of course, there's the question, what's the chicken, what's the egg?
Mm.
But in any case, there's accumulation of alpha-synuclein, and there's also dysfunction of this degradation pathway.
Okay.
So in that sense, it would probably be an idea to try to tackle both. Yeah, decrease the production of alpha-synuclein and enhance the clearance at the same
And,
time.
and one of the questions I've always had about this is, one of the curious things about these conditions is not all cells are affected. So all cells produce, presumably, this abnormal form of alpha-synuclein, whatever it is. All these cells contain this machinery. Is it that some brain cells have a better system for clearing protein than others? Or is it that they produce more of the protein? So why do the dopamine cells die off? Why don't the thing I'm always obsessed by is the visual cortex, right at the back of your brain, very active. All of it's active now as we're staring at this screen. You have these cytochrome oxidase, metabolically very active areas, but the visual cortex just seems never to die in disease processes, well, rarely in neurodegeneration, whether it be Parkinson's or similar. So why do certain cells die? Is it to do with the fact they have different autophagy machinery within each cell, or?
Yeah, that's fair. I don't think that's has been answered, comprehensively. But
Yeah,
there are, for example, some data that show that, there are differing levels of alpha-synuclein in different cell types.
yeah,
And it seems that at least in the models where you inject, fibrillar forms of alpha-synuclein, you see how they spread out into the brain and what damage they do to certain areas of the brain, that a lot of the damage relates to how much alpha-synuclein is expressed in the particular region. So
okay,
that's certainly one factor. But the other factor you mentioned, actually, we're thinking about addressing that more comprehensively in a grant application to really look at, deferring levels of, endogenous protein degradation mechanisms in different
yeah,
neuronal subtypes to see
okay,
perhaps that's explains some of the regional vulnerability.
okay,
We've already done some experiments where we've downregulated the CMA pathway in various areas of the brain, and we see that, for example, if we downregulate CMA in the hippocampus, it's not as damaging as when we do that in the nigra.
okay.
And although alpha-synuclein does
Damaging in terms
accumulate,
of alpha-synuclein aggregation and cell death.
yeah, there's no cell death when you do that in the hippocampus. There is some accumulation of alpha-synuclein, but it's not to the amount we see in the nigra. So
Okay,
it seems to be a more sensitive system in the nigra.
okay. And do
And
they have comparable levels of alpha-synuclein? Because you were saying that's the other critical factor.
yeah, yeah, pretty much. I mean,
Yeah,
they express quite a bit. This is in the
yeah,
dentate gyrus
okay.
of the hippocampus. Yeah.
And I'm just gonna this will be the final question, and I want to just quickly move on to your role as the program chair of the WPC. Uh, I mean, obviously, this is a fantastically exciting area, and it comes back to a question we had earlier. Is there a worry that with this pathway, you will just churn away, you'll get rid of the alpha-synuclein, which is causing disease, terrific, but you'll get rid of wild type, normal alpha-synuclein, so your synapse doesn't function quite so well. You'll get rid of a bit of tau, which actually makes your nerves communicate quite well. And is there any data to suggest that if you upregulate autophagy A, it can only be upregulated to a certain point and then it seals itself off? Or is it actually damaging? Is there any evidence that you've upregulated autophagy to the point where cells get exhausted, die, or whatever?
Well, there is some data that you can excessively, for example, when in some models where we see damage to the CMA pathway, the chaperone-mediated autophagy pathway, for example, for mutant alpha-synuclein, the pathway is blocked.
Yeah.
Then macroautophagy, the other autophagy pathway, is upregulated, and perhaps in some such settings, that can be damaging.
Okay.
But so far, with our approaches of chaperone-mediated autophagy induction, we have not seen any
Yeah.
toxicity, including in the primate model I mentioned.
Yeah.
So there are some models in which you do see detrimental effect of overactivating at least macroautophagy,
Yeah.
the other form of autophagy, which is more bulk autophagy,
Yeah.
and potentially also in the nerve endings in the axons.
Yeah.
Um,
I
it
mean,
seems to have a certain balance. Either if you have too much or too little
yeah.
autophagy, this could be damaging.
Yeah.
So there's
And also the
certainly
other,
a window, I
yeah.
think, a
And
window you
also
have
it's a
to operate
pathway,
in.
isn't it? So I've always said this, it's a pathway which starts and ends. And obviously, if the problem's at the end and you start up regulating the front of it, you're going to
Yeah,
make it worse because you're not targeting where the obstruction.
yeah.
But
And I
it's
think the same is true for alpha-synuclein, although, in experimental models, which are, for example, very genetic models, which I think are the best models we have, but also in humans, there are some humans who genetically don't have alpha-synuclein, and
yeah,
they are fine. So
yeah,
I don't think that there's any danger in my mind, at least in adults
yeah,
brain, to downregulate at least, 50%, for example, alpha-synuclein.
yeah.
And there's no genetic evidence that this is, damaging in any way. So
Okay.
I'm
I
not
just sort of,
concerned about that, being a problem.
so one quick question before I come to WC. Are there any people in the Contursi family who carry the mutation who haven't developed the disease and have lived to a ripe old age? So are there people who are resistant to this mutation?
Yeah, I've mentioned this in my blog, but for me, this has been a tremendous kind of life experience.
Yeah,
Actually, after Colombia, when I came back to Greece, I first these research interests I had, I joined this research institute, the Biomedical Research Foundation of the Academy of Athens. It's a mouthful. Um, the academy. And there I continued doing lab work, but I eventually, got a position in the university, also a clinical appointment, started seeing again patients with mainly with Parkinson's disease and other movement disorders. And that's when I saw this lady who was from one of these families. And then I started seeing, one led to the other,
yeah.
and we have, tens of subjects we follow from these families.
Okay.
It's a very unique life
But
experience.
some of them
We
seem
visit them
resistant.
at their homes. Many of them live in remote areas of mountainous regions in Greece. And it's very difficult for these families because the disease hits every generation and at a very productive age.
Yeah,
So the healthy members of the family have to take care of the. It's
yeah,
like Huntington's disease in a sense.
yeah,
You
yeah.
know, you
But
have
there are
seen
no
that with
members of these
Huntington's disease.
families that are who are resistant to it. So despite carrying
Yeah.
the
So within these families, we've seen it's the penetrance, what we call it. So having the
yeah,
mutation, but what is the chance you'll develop the disease? It's about 80 to 90%, but it's not 100%.
okay,
And we had this famous 90-year-old woman who never developed the disease. Her four siblings had the disease,
yeah.
but she never developed it, even though
So
she
these
had the
may be
mutation.
very instructive, like some of the
Yeah, and if
amyloid
we knew
patients.
what protected her, we
Yeah,
may have, a cure,
yeah.
but we don't know yet.
Yeah. Well, we're almost out of time, and it's been a fascinating story about alpha-synuclein, its discovery, how it lies at the heart of Parkinson's disease. So I assume this is going to be in Phoenix. We're going to hear about more
Right.
about alpha-synuclein in Phoenix. And in the last minute, Leo, just tell us a little bit about Phoenix, how you've put the program together, and what that role involved as the program chair.
Yeah, we've been working on this for a year. So it's slowly coming together. We have the program, not the final program, but almost the final program of the main lectures, and we had a great meeting recently in New York, Brooklyn, I should say, Brooklyn, for Eli's case.
Yeah.
And it's a great atmosphere. I've many of the people I hadn't met, especially I did not know them, especially from the allied health field, and I think it's great also to combine all these different interests people have, and try to bring something together that's for interest for the whole community, and
Yeah.
it's coming together very nicely.
Yeah.
And I'm
Well,
very excited about it, and I'm very honored to be part of it.
yeah. Well, I want to thank you on behalf of the community for taking on that role. I also want to thank you for a fantastic, conversation we've had today about your sort of pioneering work in this and the way that this whole field has evolved. So thank you very much. I'm afraid we're out of time, but I would like to thank Supernus Pharmaceuticals again for supporting this series, without whom we wouldn't have this. We have another one of these in April, Tuesday, April the 15th. Andrew Sideroff will be talking about clinical trial design. So I think that will be of great interest. And just as you've already heard, do not forget, you cannot forget, that next year in May 24 to 27, Phoenix, Arizona is where all the action will be when we have our next WPC. I think there are links which have appeared in the chat, which you can go to, which will give you more information on it. As Leo was saying, we had a meeting in Brooklyn where the sort of final program is really coming together now. So it's looking very exciting. There'll be something for everybody. So enjoy it, and I look forward to seeing you all again in April.